Hello, this is Chris Keating. I'm a physical chemist at Penn State University. My lab does colloid and interface science, and I'm a final speaker in the prebiotic complexity module of this PCE3 virtual workshop. I want to start by thanking the organizing committee for all their efforts in putting this workshop together, and also a big thank you to the other speakers. I've really learned a lot watching their talks, and I know we all appreciate the effort they've put into them. It's been great talking to everybody on Friday afternoons as well, getting a lot of fantastic new insights into how people think about this area. I'll be talking some about the processes that come after prebiotic polymers are available, focusing on the assembly and compartmentalization of these molecules. This is a large and active field, and what I'll be able to tell you today is just a small taste of what's being done, and I hope will help people outside these areas to get a flavor for some of the ways people have approached assembly and complexification. I apologize in advance for all the fantastic work that I had to leave out of this talk. In the earlier talks, we've already been introduced to oligomers and polymers. Once these molecules are present, we can start thinking about how they fold into three-dimensional structures and how they interact with each other into assemblies, which can become compartments for prebiotic chemistries. You should be picturing things like RNA folding, DNA duplex formation, and lipid self-assembly to form vesicles here. So let's take a moment to think about what's driving self-assembly. Fundamentally, assemblies are driven by enthalpic and entropic processes that include a variety of attractive and repulsive interactions, including also counterion release and solvent reorganization, which are more sort of entropic in nature. Shown here are some of the ways that molecules interact. And on the right, I want to call your attention to the fact that these interactions respond differently to solution conditions. For example, ion pairing interactions shown at the top between cations and anions are much more sensitive to solution ionic strength than pi-pi stacking interactions shown at the bottom. Other important interactions that I want to particularly call your attention to are metal coordination and hydrogen bonding. Hydrogen bonding is a really big deal in aqueous solvents and is responsible for the hydrophobic effect, which is the tendency of oil and water to segregate. And this is understood as an attempt to minimize the entropic cost of messing up the water hydrogen bonding network. So here are some familiar examples of molecular self-assembly. RNA folding, which involves different parts of the same molecule interacting intramolecularly to form a three-dimensional structure that can give rise to catalytic function based on how the functional groups are coordinated in space. Nucleic acid base pairing, intermolecular hydrogen bonding, and base stacking interactions can provide sequence-specific complementarity, and that's really important for replication. You're seeing a sort of close-up of it down here, where these differences in how the bases come together allow them to recognize each other in solution and select the right one out of a mixture. Another example is the hydrophobic collapse of proteins, in which nonpolar amino acid side chains are buried in the interior of globular proteins. And I'm showing sort of a cartoon, but then also a chop through a native protein. This is lysozyme. We're going to chop it right there. If you look inside, these red labeled residues are the hydrophobic ones, and they've been buried on the inside. Self-assembly, of course, is not limited to molecules important in extant biology. There's an example of a non-covalent supramolecular polymer. And this sort of polymer has been hypothesized to predate modern nucleic acids. So you can see it takes advantage of some of the same types of interactions along a polymerization that's in fact not covalent in this direction here. So a variety of molecules can undergo self-assembly, not just limited to the ones here, but just about any molecule can have some sort of self-assembly occur. I know we've been talking a lot in the virtual workshop about what types of environmental conditions are likely or necessary. And I wanted to say a few words about that with respect to these sort of familiar self-assemblies of molecules like DNA base pairing. So this is a plot came from the website of a company that sells DNA oligonucleotides, and they help you figure out the sequence that you want to design and purchase. And it's showing the stability, the transition melting temperature, where you go from a duplex to two single-stranded DNAs, happens at a particular melting transition temperature. And this is being plotted now versus the concentration of potassium and of magnesium. So looking at a monovalent and divalent cations, this is divalent, even though it's shown with just 1+. And you can see that certainly it's very responsive to what the concentration of these cations is in solution, but that you can sort of make up for the lack of one by having more of the other. And this is something that I really want to sort of focus on. Sometimes when we do self-assembly, we can compensate for an adverse environmental condition if we can vary some other condition. For example, here, if you don't have much magnesium, it's okay as long as you can have a lot of sodium, or potassium is what's shown here. Sodium would work too. And those kinds of trade-offs are things that we're routinely able to make in self-assembly if we know what the conditions that are really fixed and which ones are variable are. If we're thinking about prebiotic environments, that means that what's really helpful to know is actually a set of linked conditions that might occur together versus which other conditions might not ever occur together, because that will help us to figure out which things we can change to make up for other deficits sort of in the solution conditions and which things we're really stuck with. And we have to change instead our molecules composition in order to cope with those. I might have a chance to come back to this later in the talk. So another way that self-assembly is interesting and important, besides forming catalytic sites and forming duplexes that can give us sequence selective replication, is you can actually just prevent molecules from going away. So prevent degradation of molecules by, for example, hydrolysis. And this is actually a pretty general thing that's been observed in a lot of the structures we saw on the last page, but also can be observed in some sort of heterostructures between more than one kind of molecule. And this is a really nice example from the recent literature earlier this year with deoxypeptide RNA duplex combinations, where you have a free RNA duplex, and under the conditions of this experiment, it's not stable and it will melt into the two individual strands. There's an intact deoxypeptide, and under the conditions of the experiment, it's not stable and it's going to hydrolyze. And so those are both unstable, but when they get together, they bind. And that complex of the two types of molecules is stable and persists in solution. This is not unique to RNA duplexes and deoxypeptides. Here's a completely different system where lipid vesicles are being stabilized by prebiotically relevant amino acids, in this case, serine, although the paper looks at other sequences as well. So this is with no amino acid. We see sort of dimly some vesicles. When the serine is there, they actually get sort of brighter and thicker looking, the lipid fluorescent label. When you expose these to various salts, which are destabilizing for this particular kind of assembly, what you see is that these don't look so good anymore in the no amino acid case. They look less like they can have something inside. And the serine ones stay sort of vesicle looking. When they looked at these under the cryo-electron microscope, what they saw was that the number of vesicle layers that they saw without the serine added versus with was different, that we had more lamella, more lipid bilayers in the serine containing one, and that was aiding in their stability. So this is a sort of general approach that we see across many types of molecules and many types of assemblies. That actually brings me to the next thing I wanted to talk about, which is the formation of primitive compartments by self-assembly. We had a really nice introduction from Dr. Diemer earlier in this session about lipid vesicle formation and how these prebiotically relevant compartments were a plausible container for prebiotic chemistries. Lipids can self-assemble into vesicles with semi-permeable membranes, similar to those of modern cells. Vesicles have been a major focus of prebiotic compartmentalization research, and many important capabilities have been demonstrated. A second type of compartment that people may not be quite as familiar with is a phase separation that forms something called a coacervate, which is a liquid droplet. And so it's an alternative solvent that's formed in the initial solution. These structures are also common in modern biology, and they've only been recently appreciated to be a mechanism for forming a host of membraneless organelles that occur in both the nucleus and the cytoplasm of eukaryotic cells and also in some prokaryotes. I'm showing a general phase diagram here. It's this guy, where we're looking at the concentration of polymers on the x-axis and then weakening the interactions between them on the y-axis as we go up. And in this region marked in blue, that's where there's going to be a second phase appearing. Everything will be soluble in one phase out in the white here. And you can read a lot of information into the phase diagram. They mostly have this sort of shape for coacervates, but the details will vary with the system. And then you can see some compartments shown down here, and we'll come back to things like that. One important thing to note is that while liposomes have an aqueous lumen, where if you put an RNA or another molecule inside here, you can expect it to behave about the same way that it would when it was outside the vesicle, right? If it was folded and catalytic outside and you put it inside, it should still be folded and catalytic. If you do the same thing with coacervates, your mileage may vary, because inside of a coacervate, it's not just water, it's all of these polymers, right? It's a whole entirely different solvent environment. And things may be very different. Your molecule may or may not fold the same, it may or may not be active the same. That can be good or bad, but it's important to note the difference between these two types of compartments. So compartments generally are really important on the pathway to life from prebiotic chemistry. Forming compartments is one of the things you really need to have advanced prebiotic function. Some of the things that they could do are listed here. You can maintain high local concentrations. This might allow you to shift the equilibria to favor molecular associations that might not happen in dilute solution. Might allow you to have an enhanced rate, co-localized molecules that might not otherwise be together so that they can interact. It can also provide favorable microenvironments. So the inside of the compartment may have a different pH. It might have a different ion concentration, local solvent properties, etc. It might protect from damaging conditions or reactants. And if you have a semi-permeable boundary, you can control which comes in and which comes out, which will help you to pre-concentrate reactants, for example, or to remove products more efficiently. If having a compartment changes the rate of a reaction or turns on and off a reaction, then if you make the compartment come and go, that's a way of controlling that reaction. And so this can have a regulatory role. Finally, with a particular note towards prebiotic chemistry, you can define individuals by having a compartment. Once molecules are together in some sort of vesicle, perhaps in a coacervate, this is now a structure that selective pressure can act on. And so this is going to be a really important step in moving from collections of molecules that are just each hanging out doing their own thing to something that eventually could be a protocell. So I mentioned lipid vesicles and coacervates as compartments, but I also want to stress those aren't the only compartments that are possible or that are viable. They're the ones that are the most self-assembled, I suppose, and so I'm focusing on them in this talk. But I want to call your attention to the fact that A, there are lots of other things that could be compartments, and B, one of them is aerosols. And in aerosols, you actually have a combination of sort of a physical mechanism where wave action sends water droplets up into the air and the atmosphere, and then also molecular self-assembly, where molecules that are in that water, perhaps because they were at the air-water interface, perhaps they're a little bit nonpolar or surfactant-like, those now are concentrated as the droplets dry, and they can form a film at the surface, they can undergo phase separation, all manner of things can happen. You see some of them depicted here. And this also is happening in an environment where not only is there dry, but there's more access to sunlight. Those then can deposit back into the ocean and do it all again. This is a close-up of what sort of a kind of a reverse micelle situation could look like for an aerosol. When a structure like this comes back to the water, it could even pick up another leaflet of surfactant and become something more like a vesicle shape. So these are actually really richly interesting compartments as well. And there are other things that could be considered here beyond these. I'll focus on the ones that are mostly self-assembly going forward, but just want to draw your attention to the fact that that's not the whole set of all compartments. I want to say a few more things about lipid vesicles. These compartments have semi-permeable boundaries that in general can retain large or highly charged molecules, like RNA, for example, and allow smaller, less charged molecules to pass, water, sugars, even activated nucleotides. The molecular structure of the lipids making up a vesicle determines its permeability. The more prebiotically reasonable single-chain lipids forming more permeable and less stable membranes as compared to modern 2-acyl-chain phospholipids. This is encouraging because modern cell membranes require complex transmembrane transporters that will bring things from one side to the other, often with the input of energy. However, much simpler peptides are also able in simpler systems to sometimes transport, for example, ions along a concentration gradient. Importantly, it's possible to load reasonably high concentrations of solutes into lipid vesicles by approaches such as wet-dry cycling that concentrate the molecules to be encapsulated while membranes assemble. As we heard in the first talk of this session, I took this nice picture directly out of Dr. Diemer's talk, sort of catching the idea that if you have these little solutes and you wish they were inside the vesicles, or perhaps they're in some sort of prebiotic pond, then drying will make this a multilamellar lipid matrix that will trap those molecules inside of it, such that when it's re-wet, the vesicles will form and encapsulate them. And this will give a much higher encapsulation efficiency than if you were to just form the lipids in a dilute solution, like the one that you had initially. So this is important for encapsulating molecules at reasonable compositions inside, concentrations inside. A variety of other really impressive attributes of lipid vesicles have been demonstrated over the years, including, for example, the ability to discriminate between simple sugar molecules that you would think would pass the membrane sort of with similar permeability because they have similar sizes and chemistries and hydrophobicities. But in fact, there appears to be ways for the membrane to nonetheless select for some over others. For example, ribose is especially able to permeate the membrane. Another important thing that's been observed for lipid vesicles is their ability to grow by addition of new lipids from solution. And that's really important for the idea of transitioning these into protocells that could grow and divide. These observations and a host of others that we don't have time to go through today suggest the possibility that vesicles could serve as protocells. They could house catalysts, information, and or metabolism. And so you can think about how you would envision such a protocell. And here's an example of one that's been envisioned for sort of an RNA world scenario, where we have a relatively simple permeable lipid membrane that's encapsulating an RNA. And it's allowing the monomeric precursors to pass the membrane. And it's growing by adding lipids from solutions, such that the RNA replication and the vesicle division are coupled. One thing that's nice about lipid membranes is that their composition can change over time. So as new lipids become available that have different structures, they can incorporate in an existing membrane. And changes in the composition of a membrane over time could reduce the permeability of the membrane. For example, if these single chain amphiphiles started to adopt more and more double chain amphiphiles, such as that are more similar to the modern lipids, that adds to their stability and it reduces their permeability and ultimately moves them closer to the kind of membrane that we have today. So it's an interesting way to think about sort of compositional change over time that could be selected for and lead to new functions that are more appropriate for later types of structures. So to understand how molecular structure influences the physical properties of membranes, it's instructive to look at the relative sizes and shapes of the hydrophilic and hydrophobic parts of the molecules. Here I've highlighted in green the acyl chains of a more prebiotic single-chain fatty acid and also a modern diacyl phospholipid. And you can see that the shapes of these molecules are really quite different. Physical chemists like to look at the shape in terms of a critical packing parameter. Basically, you can see this on the right-hand side of this figure. And these will form these characteristic assembly types that you can predict based on the shape of the individual molecule. Cone-shaped molecules will form micelles, while cylindrical ones form bilayers, which are needed for vesicles. Anything that changes the molecular shape can change the assembly. For example, this fatty acid is shown in its deprotonated anionic form. But if the pH is lower than the acid dissociation constant for the carboxylate, it will become protonated and then be neutral. That will reduce the repulsions and make this molecule change from a cone shape to more of a truncated cone. Similarly, if the ionic strength is raised by adding salt to the solution, that will screen the charges and allow the fatty acid head groups to pack more tightly, even if they remain charged. Everything matters for prebiotic encapsulation by fatty acids. Here's data for oleic acid as a function of pH. As you change the solution pH, you change the protonation degree, and that switches you from micelles to bilayer vesicles to oil. The bilayer vesicle situation, shown here in a TM picture and here in a cartoon, requires that you have a mixture of the deprotonated and protonated forms of the lipid. And this is shown schematically here over on the side, where at very low concentrations, all these lipids just exist individually in solution, but above their critical micelle concentration where they can assemble, then it depends on the relative location of the pH to the pK, the acid dissociation constant of the molecule, where if you are above it, you have anionic head groups. If you are near it, you get this mixture of anionic and neutral head groups, which is what we're going for here to get vesicles. And if you are below it, then you get neutral head groups, which in this case makes the molecule insoluble and it phase separates as oil. So compartments formed by phase separation were proposed as protocells long ago and have recently been having a resurgence due to the realization that they are common to living cells and are important in numerous cellular functions. Coacervates formed by associated phase separation of molecules can happen in a couple of different ways, depending on what those associations are driven by in terms of their molecular interactions. I'm showing two sort of limiting cases here in cartoon form. One is a complex coacervation, where there's complexation between a cationic and anionic molecules, and these need to be multivalent and weak interactions for it to form a liquid as opposed to a solid. You can also have what's sometimes called simple coacervation, which is when it's not driven by ion pairing, so you don't make a complex. These are often driven by changes in solvation, which are temperature temperature driven, but it can be driven by other things as well. This is now a picture showing some droplets. This is a single liquid phase that's formed out of another phase. So we have two total phases. The droplet phase is the coacervate phase. Outside of it's what's called the dilute phase. It's sometimes called the supernatant phase. And the reason for that is if you spin these things down in a centrifuge, it will be the supernatant. And I'm showing that here. This is the top of the supernatant phase and a little Eppendorf. And here is the coacervate phase. So all of those droplets now have been joined into one. They coalesce when they touch because they are, in fact, just one phase that's been separated. They're not coalescing here because they're sitting on a glass slide, and so they're not interacting with each other. They don't know that they could get together and coalesce. In the same way that if they were sitting on a mineral surface, they wouldn't have been able to coalesce if they don't touch each other. So what drives accumulation of solutes into a coacervate? Sort of a different process than what happens for lipid vesicle loading. Solutes can be accumulated into these droplets by partitioning. So this is basically the solute gets to decide where it prefers to be based on its equilibrium partitioning coefficient, which is the difference in delta G for it being in the two different solvents. And that's what's being sort of shown here. And what your solute may prefer might be to be inside, it might be to be outside, or it might have no preference whatsoever. What dictates that usually is going to be interactions with the components of the coacervate. And those are illustrated here, some of the main driving forces, whether there's hydrophobic interactions, for example, of this molecule in yellow with parts that are in the coacervate already, whether it's charge-charge interactions, that's quite common for complex coacervates, whether it's hydrogen bonding, for example, there's already a molecule in there that it can hydrogen bond with effectively. Often you also have influences in partitioning based on whether the molecule can effectively enter due to sort of geometric concerns. So these coacervates do have kind of a mesh-like structure where there's transient interactions, but they're between these large polymers. And so infinitely large molecules sometimes can't enter effectively, and that's what they're trying to show here with this mesh. And finally, a replacement mechanism is actually quite common in complex coacervates, particularly for polyanion solutes like RNA that we tend to care a lot about in prebiotic chemistry. In this case, if an RNA gets accumulated into a coacervate, it's usually because it's kicking out whatever anion was in there before. And so whether it accumulates really well or really poorly has to do with its relative goodness as an anion compared to the one it would need to kick out. So if it's longer and more charged, it's more likely to be able to displace that existing anion. Also can depend how big and how high charge density the cationic component is. Because if the cation can't perceive a difference between the two anions, it won't show up in the partitioning. Much of the work so far has been done with complex coacervates because their interactions are due to ion pairing. That makes it pretty intuitive to figure out what's going on in these systems. And so the literature disproportionately discusses complex coacervate partitioning, but the driving forces are, of course, not going to be replacement or ion pairing for the other type of mechanisms for coacervate formation. And one of those is shown here. This is exciting work recently done on a prebiotically relevant system where these alpha hydroxy acids existed as monomers, and they were dried slowly at 80 degrees, a process that led to their polymerization. And as they polymerized and dried, then when they were re-wet later, they formed coacervates, and those coacervates are shown here. These coacervates were able to accumulate dyes or RNAs, similar to other coacervates. And mixtures of different alpha hydroxy acids were also able to form droplets that could function as these fluid compartments and concentrate solutes such as the dyes or RNAs. And so it's sort of exciting to have the combination of the oligomerization and compartmentalization happening sort of all at once here, particularly in a system that is starting to get a little bit messy. So that's nice. Thinking about environmental effects on phase separation, the two different systems that I outlined before have different reactions to changes in environmental conditions. And so I'm showing here on the left some complex coacervates. And again, ion pairing is very important. And charge screening due to added salts can prevent their formation. So we're comparing here different repeat lengths of peptides in complex coacervates. And we're looking at the turbidity. So when they're turbid, that means there are coacervates present. When the turbidity goes away, That means the amount of salt present prevents them from forming a coacervate. And as we move from the low salt to high salt stability, what's changing, if you look at all these different symbols, is the length of the polypeptides. The longer polypeptides can survive in higher salt conditions. So this red curve here is 100 MERS, which is not super prebiotic. And on the bottom, we're looking at arginine aspartic acid and arginine glutamic acid as opposed to lysine. And these are actually much more effective at surviving to high salt. In fact, this green curve is only for 10 MERS. And so it's because the binding interactions that drive formation of these are not as cleanly ion pairing. There's a lot of cation pi and pi involved in these coacervate formations. And so you see that in their different response to the ionic strength. If you move to a different mechanism entirely, though, so these are elastin-like polypeptides. So they're like a protein, tropoelastin, and they have these repeats with relatively hydrophobic residues. And when you change the repeat sequence, you change their phase separating behavior. And what we're looking at here, as the absorbance goes up, that's also at the top coacervates, at the bottom it's not coacervates. And what you're seeing as a function of temperature is that based on small changes in the sequence here, they're able to make coacervates appear as they got hotter or disappear as they got hotter, and they can shift that temperature around quite a lot. It does depend on the length, the number of repeats in addition to the sequence. And these are okay, relatively long sequences, but one thing I do want to point out about them is that the peptides are, they're not homopolymers, which is nice, and they're somewhat hydrophobic. More of these amino acids might have been prebiotically relevant than, for example, a homopolymer of arginine. And when you start to add high concentrations of salt, that actually helps them phase separate instead of hindering the phase separation. So what I really want to stress here is the effect of environmental factors like pH, temperature, salts, et cetera, are going to depend on whatever the mechanism was of phase separation. Generally, they're going to be tunable within some window of conditions by changing things like molecular structure or repeat length, or by changing some other solution condition that can help make up for the one that's driving against coacervate formation. Something that may not be sort of obvious that I want to point out about coacervates is that they can discriminate between molecules that seem similar. So like single-stranded and double-stranded RNA, or single-stranded and double-stranded DNA. And this is an example from literature with single-stranded versus double-stranded DNA, where the single-stranded forms formed liquid coacervates, but when you had hybridized double-stranded DNA, they formed instead precipitates. And one of the reasons that this seems to be true is that the single-stranded ones have a shorter persistent length. They're more flexible, and they're better able to make the sort of type of weak transient interactions than the double-stranded DNAs. This type of distinction between double-stranded and single-stranded nucleic acids isn't just about forming coacervates. It's also about going into a pre-existing coacervate that's made of something else. And this is a kind of complicated picture. I apologize for that. It's a really cool paper, though. And what they've done here is they've taken an intrinsically disordered protein from modern biology, which makes coacervates inside cells, and they've asked what happens when you put different lengths and double-stranded versus single-stranded of DNA and RNA. Do they go in or do they stay out? And the blue ones here, they all go in. Those are all single-stranded DNA or RNA. And these other colors, the short ones of double-stranded, they go in, not as well as the single-stranded. The longer ones, the longer double-stranded, are excluded from the droplets. And so the concentration inside is lower than the concentration outside, whereas the concentration inside of the single-stranded is much higher. Interestingly, double-stranded short things like these hairpins were able to get in just fine. And they interpret this in terms of a mesh-like structure that can't handle the high persistence length of these longer duplexes, so 20 MERS and longer. That kind of discrimination is kind of interesting. And they also observed that the fraction of duplex present on the inside of a coacervate was lower than on the outside. So it seemed like it had an effect of essentially melting, if you will, the duplex. And you may ask yourself, well, this is some modern protein. This is probably not a general thing that would happen somewhere else. But in fact, we've seen it in some of these simple polypeptide ones as well. under certain conditions. And so it seems maybe not just something specific about this protein, but maybe more something general about the structure of coacervates, their reduced polarity, and their mesh-like structure. People have thought about how you might draw cartoons similar to the protocell liposome RNA replicator that we saw earlier. And this would be a cartoon like that, where you're thinking about a coacervate that concentrates nucleotides and magnesium, and that facilitates their oligomerization and non-enzymatic replication. And as they get longer, they're more effectively accumulated, and other ones are competed out, and they can fold and then become catalytic. So some parts of this have been kind of experimentally validated, and other parts we're still needing to learn more about. But people have put some RNAs inside of coacervates, ourselves and others. And one example that I'll show here is sort of relatively simple, where we start out with this hammerhead ribozyme. It's in these coacervates formed of these molecules. And at a relatively high concentration of the RNA, there's no difference between the coacervate and the buffer. So it's active in the coacervate. It would be just as active if there are no coacervates. It's not really gaining you anything to have the coacervate there. But if you reduce the concentration of the enzyme, it doesn't work anymore in the buffer because its kinetics are concentration dependent. And if you have the coacervates there, it's able to concentrate that RNA inside, and it doesn't take nearly as much of a hit to the reaction rate. These types of experiments have been seen in a couple different kinds of coacervates and now by a couple different labs. So there can be ways of rescuing activity by encapsulating within a coacervate. But like I mentioned before, what you're doing here isn't just concentrating it, you're also putting it into a different solvent environment. And so your mileage may vary, right? Sometimes it actually slows down rather than speeds up. It depends on the composition of the coacervate and the environmental conditions. Most experiments have been done with simple, well-controlled mixtures of just a few molecular types. So they're possible to interpret. We do that on purpose generally, even though we know real life is messier. But as we've been discussing in this workshop, prebiotic chemistry can be expected to be not just a little bit, but a lot messier than our laboratory test tubes. What will happen when we try to move closer to that? If you add to the molecular diversity, that's going to be really important to the selection of chemical evolution of assemblies and compartments. And it's something that researchers are increasingly excited to embrace, like in learning or trying to learn how to embrace effectively. So I have on this slide two really still very simple baby steps that have been taken towards increasing complexity. Where on the left here, this is now instead of adding one polycation and one polyanion, there are three of each. And instead of getting one kind of droplet, we get, I don't know how many kinds of droplet this is. There are more than 3 coacervate phases. And if you stare at it long enough, it's disturbing how many different things seem to be in there. But maybe we shouldn't be disturbed. We should think about this as a population that could be selected. On the right hand, we see an example where fatty acids were added to coacervates. And the fatty acids formed a sort of thick multilamellar membrane around the outside of the coacervates. And this altered the uptake of other materials into the coacervate. So it's starting to kind of provide a little bit of a semi-permeable boundary around this crowded, concentrated coacervate that sort of starts to become now a protocytoplasm. And this is an exciting step towards hybrid protocell models. We think about some of the things that the different kinds of assemblies might have particular effectiveness in making happen. And if we can bring them together, we might envision protocells that would benefit from both. So as we think about this transition between assemblies and protocells in the first life, it's important to consider roles for changes in environment on compartments themselves and on protocells that they hopefully sort of grow into. These could be cycles like wet, dry, hot, cold, day, night, or they could be gradients, for example, around a hydrothermal vent. These spatial or temporal changes in environmental conditions could apply selective pressure to compartments and to populations of compartments. They could disappear, the compartment could persist. It might have a different composition or it might have a different function as it moves through these different environmental scenarios. So I have a couple examples at the bottom here One in which a self-reproducing liposome protocell over here is experiencing changes in pH as a selective pressure, and another on this side where wet-dry cycling of coacervates is releasing and recapturing an encapsulated RNA oligomer. There's countless other ways that such changes in conditions over time or space could act upon assemblies and compartments and protocells. So here we're seeing a hypothesis for how this type of selection could lead to chemical evolution of protocells in an inland hydrothermal site. And start out with this hydrothermal site, sort of loading up some vesicles here. And these vesicles then are encapsulating some sort of system of molecules within them that's then acted on by the environment. And some of them are being disrupted and their lower stability makes them go away, and the others persist. And this can then can cycle. So these protocells are carrying encapsulated molecular systems. And generally we think of these encapsulated molecules as the information carrying part of the system. But it's also possible to think about the molecular composition of the assembly itself as information, compositional information. And that's what's being illustrated here, is this idea of the actual composition, for example, of this vesicle, my cell, I guess, is the information. And this may seem like a less effective way to carry information. What this model is arguing is that, in fact, if you have very high molecular diversity, you can do as well with compositional information as you could with sequence information. And so if the situation is very, very messy, that could, I suppose, benefit in the sense of having a lot of information contained in the composition of the assemblies. So I tried to give you a peek at some of the processes and observations, hypotheses for prebiotic assembly, compartmentalization, and complexification. To summarize, assembly depends on molecular structure and environmental conditions. Assemblies stabilize molecules and provide new, potentially functional environments. Assemblies can form compartments, become individuals. Synergies between molecular classes, for example, peptide, nucleic acid, and lipid, and assembly types, for example, vesicles and coacervates, are increasingly recognized in an interesting area for the future. In closing, I want to leave you with a couple of general thoughts. First, that there's growing appreciation of the importance of complex molecular mixtures and system-level thinking. Second, that despite the importance of environmental conditions, no one set of, quote, best, unquote, conditions can be described. It depends on the molecules, the assemblies, the processes, and importantly, the different conditions that can't maybe be decoupled. What are the niches that protolife could occupy as defined by early Earth environments? These conditions need not exist over entire planets, and it would be valuable to have an idea how the conditions within a given niche or microenvironment were linked, or if they're not linked, if they're independent, and what gradients and cycles are reasonable for those locations that might drive selection. Thank you.